Basically, I guess just to kick things off, so there's a couple of recent wins that might be of interest to people, that kind of recent victories a testbed can kind of notch under its belt. So a couple months ago, so Ronak had an externship in the testbed that they thought was very, very successful, and we announced that as part of PTRME. As well, more recently, back at the very end of last year, Miltenyi installed their first upgraded spatial proteomic multiplexing Maxima system microscope in the United States. So that was pretty cool. We got it first, and still the only ones who have it. Even more recently, back in I think at the end of January, Ramona Optics launched the new model of their multi-camera array microscope, their MCAM, based directly on testbed feedback, because they were beta testing it based with the help of a couple of WFirm PIs in the testbed, and so now they finally went to market with it. And actually, I'll give you another update on them later, but there's going to be a round 2 of that. And most recently, so Apredia just got announced FDA approval for their clinical use of their E1000, their slide scanner, in the testbed, thanks in no small part to the clinical trial that the testbed conducted using that scanner. Thank you again to Dr. Marini and to Katie for helping out with that a lot and basically running it. So these are basically like, feathers in the cap of the testbed, but looking forward a little more, to kind of spread the word, spread the gospel, we have this kind of comprehensive marketing strategy for the testbed that includes both online and in-person materials to just try to get these in front of people's faces as much as possible. They're all interlinked, right? So we have a testbed advertisement one-pager that's linked to a website with a direct funnel into our system that lists all the equipment and all the capabilities of the testbed, including a virtual tour, like a Google Map style virtual tour with some of the I guess most kind of gemstone equipment highlighted that people have already responded well to. So basically, our call to action here, because I can send them out again. I've sent out the website and the virtual tour and the one pager a couple times to folks, but we just want to get these in front of as many people's faces as possible, nationwide, worldwide. So everything's operational. We built the funnel. Now we just have to wait for them to come. So yeah, just get this on your LinkedIn, on your socials, at conferences, blast these out everywhere. Please be annoying about it. Additionally, kind of this is more internally, but as we try to market the testbed more locally, we have these equipment demonstrations where people come and again show typically audiences of mostly WFirm researchers what they can do in the testbed and what the testbed can do for them. Just a couple of recent ones that are worth mentioning. Biorad demoed their ChemiDoc gel imager. Sartorius is actually earlier this month, last week, had like a user showcase for how to use our IncuSites, and they're going to follow up next week with more kind of in-depth office hours with testbed users to try to increase usage as well. A potential new company, Cell Microsystems, is demoing 2 different pieces of equipment that they're going to be putting in a testbed for about a month and a half. To get WFirm users to use them. And then FetTech is also planning with the testbed a showcase for their ECM material later in the spring. But again, so if you have the ideas for any demos to recruit more users within WFirm or in the larger ecosystem, just please approach me and we can get you in the queue. And then speaking of that queue, so like it's just a sampling of some like ongoing experiment projects happening right now in the testbed. So Ramona, which already kind of went to market with their previous MCAM that was beta tested in the testbed, literally today, like an hour ago, they were installing an upgraded module that includes incubator capacity in the testbed for the same idea. Now they're going to like beta test it here in the testbed and eventually go to market with it. FetTech is also collaborating with Sean Murphy and a couple other WFirm researchers on an organoid fibrosis project with their ECM material. Brinter and Biospherix, the ex vivo, are going to have a collaboration that they're initializing right now. There's a new holo monitor that's been upgraded in the testbed as well. Geister Bio is planning to come at the end of this month. They're an Austrian company to seek FDA clearance for their kind of enclosed, incubated bioprinter. So we're in talks with them about where we can kind of shove that into the testbed, and they're coming later. And then on the more slightly academic side, so Baptist is collaborating with a testbed primarily on actually they're trying to digitize a couple thousand of their patient lung cancer slides. So that's like something we're working on now, as well as another another tumor treatment that they're using the Miltenyi Blaze in a testbed for. And then UNC Chapel Hill is collaborating with a testbed on one of their projects that involves also using the Miltenyi Blaze microscope in the testbed to image whole mouse brains. So basically, just long story short, there's a lot of things happening in the testbed right now. We want more things to happen. So again, any ideas, any collaborations, anything you might want to try out or beta test, please come to me and we'll try to get you in there. But with that, the real star of the show are the testbed member companies. So I want to turn the table over to them to speak to each other about what you can do in the testbed.
So we've enjoyed a really active collaboration with RegeneratOR and WFirm over the last 2+ years. We've developed a multi-camera array microscope. It essentially includes many small microscopes that we packed into a compact benchtop system to very rapidly and synchronously scan samples and wall plates. And so what we're really trying to enable is live 4D biology. And we've been most focused on working with the soccer labs and the seed labs on various types of organoid imaging. Stephen, if you don't mind just advancing a couple of slides there. I think this GIF is really helpful to explain how the technology fundamentally works. Most microscopes over the past 150+ years since microscopy was first invented have just one single objective for one human to peer through. And what we've done at Ramona is really create a paradigm shift in how you think about microscopy. And we've created this MCAM, or multi-camera array microscope. And so you can see on the right, this is our Vireo system. It has 24 cameras. And so we can massively parallelize that imaging and image a well plate in a matter of seconds to minutes, depending on how many channels you need to image in, how many Z-stacks you'd like to image in. The 3D nature of the sample plays a role there. But we've been able to help members of the testbed conduct their imaging of plates anywhere from around 30 seconds to 2 minutes for entire 96 well plates or 24 well plates. I'll show you what our system currently looks like in the testbed. So as Stephen mentioned, they've had actually 3 different iterations of our system. So going from bright field to 2 channels of fluorescence to 4 channels of fluorescence. And today we also integrated incubation into the system. And so we're really excited now about the ability to do longitudinal experiments with their biology. And if you go to the next slide, Stephen, we've essentially taken all this feedback over the last couple of years from RegeneratOR and a couple of other early access partners and commercialized this system, which we're now calling the Vireo system. So it officially launched at the Society for Laboratory Automation and Screening Conference in January. It's now available for pre-order, and RegeneratOR is one of the very first in the world to have this system, and it's been an extremely fruitful partnership. And we've received great feedback so far in the market. We've redesigned the system from the box structure you saw on the previous slide to this more sleek design, and we're really excited to be on the market for this. If you move to the next slide, Stephen, talk a little bit about application spaces. So the first is just very rapid quality control. So imagine if you have up to 1,000 plates a day that you're trying to image for drug screens, or just very, very quickly trying to get a sense of, are our samples healthy? Are they growing? Are they contaminated? We're able to do that at a scale that no one else on the market can, given that we can image plates in a matter of seconds. And what we're doing over time with the help of researchers at RegeneratOR is building automated assays. So rather than just getting the images, we're trying to get you the numbers and quantify those growth metrics, those health metrics, so you can make decisions even more quickly, basically trying to take you from images to insights in a matter of seconds to minutes rather than hours with traditional microscopes. We're really excited about live cell drug screening, being able to actively screen for many different compounds over many different wells at a time. We can also image 1536 well plates. So really just increasing the throughput of experiments and the number of combinations or permutations that we can offer at scale. And then the last is cellular dynamics. And this is the area we're perhaps most excited about because we're really uniquely suited to do this, given that we have 24 cameras. So we can park each camera over a well and we can image those wells in video rate and in time lapse. And so that's one thing that we're doing with Cecilia Schaff and Damien and several others on the soccer and Atala and Seeds teams. They're trying to watch actively T cells killing cancer cells, and they're introducing different types of chemotherapies and immunotherapies. And we feel that this is one of the only groups in the world doing this type of research, and they're able to do this across 24 wells at once. So really just increasing the throughput and reproducibility of their experiments, and hopefully getting to answers faster and getting to personalized therapies faster. So I will stop there, but really thrilled to be part of this community and very grateful to Stephen, Katie, Josh, and the whole team. So thank you all.
Thanks for the chance to share with everybody. We're probably going to hear a lot about some equipment. Pluristyx is more a software piece in that we supply stem cells. So here's my information, but we're about a 5-year-old company, 7-year-old company now, based in Seattle, Washington. We have brought in 2 technologies: the ability to reprogram cells from fibroblasts or other cell types into stem cells, pluripotent stem cells, and mixed that with some gene editing capabilities that we brought in from a merger. A company used to be called Pancella, both a safety switch type technology as well as an immune evasion technology. So what Pluristyx offers for the test bed is sources of cells, stem cells. They can be research use only. They can be clinical stem cells. They're all made with a footprint-free reprogramming, mRNA reprogramming methodology. So there's no leftover virus particles or DNA particles that would be left in the cells once you're eating them. And we can mix that with some gene editing capabilities. We can do an HLA null, knock out the B2M and the C2TA genes so that you effectively mask the cell from seeing it as foreign. Or we have a technology called IACT stealth cells. That's an overexpression of several different factors on the outside of the cell. It makes it seem more like it's natural in the cell body. And then we have a safety switch, a kill switch. It's called failsafe, and it works very easily. Cells that are rapidly dividing can be killed with the admission of ganciclovir, an FDA-approved drug, and cells that have been terminally differentiated and are not dividing would not be killed. You can use that either in the future treatment of a patient or even in the selection of cells once you're differentiating, because the stem cells would still be dividing. You could get rid of those as you've moved through your differentiation pathways. And then we have some cryopreservation services so that we can help you with freezing cells. We've got some medias and some know-hows around that we would love to offer. All of this is backed in kind of a try before you buy type business model, in that we can give you options to get vials of cells without executing a commercial license where you're spending hundreds of thousands of dollars for cells. And would just love to make these available to you. I know that the organization in the RemDo organization is figuring out ways to make that licensing even simpler for all the companies in the test bed. So that's Pluristyx. Thanks for the opportunity.
So good afternoon. Thank you, Stephen. And, thanks, Terry, and everyone else for putting this session together. I think it's been a long time coming, and we are honored as Biospherix to be one of the original members of the test bed and really thrilled to continue to promoting it, right? So I think part of my goal from today's meeting was to ensure that we understood Biospherix on our own. We do nothing by ourselves. We are the enclosure for all of your equipment, processes, and products. And so, some of the things I wanted to talk about were, in the summer last year, we did a 5-week study with Ronak that was absolutely fantastic. Katie helped with that, and Ronak sent a member here for 5 weeks, rented an Airbnb, and that is what we want to do more of, right? We've got studies that we've done in the past with Ronak. We've partnered with our friends PHI. We're working with Brinter on a study. We're working with Cellbox to do some studies. And I think one of the ultimate goals from our side at Biospherix is more collaboration. In other words, how do we put together a commercialization package to say, you know what, if you're using a particular media with a particular cell, you need a certain device to check on quality via imaging or other tools. You want to have it in an environment that is cytocentric, which we would provide. You want to have a way to transport from A to B, which we could do with companies like Cellbox. And I think that is what I hope at some point, either today or in the near future, we'll sit and think about. How do we take Steve's Pluristyx cells, show them that they work with a particular media, and make it easy for the user? In other words, I, as a customer, don't want to have to figure out what tires I need, what engine I need, what seats I need, what aluminum to make my metal, because can't be Canadian anymore, what I need. Sorry, I don't know where that slipped. But in any case, I want us to consider collectively sitting and thinking about how do we build a package together that says, look, if you're a researcher and you want to work with a cell line, we can get you a cell line from Pluristyx. We know it's been tested in this media. We know that we can use PHI imaging to test quality prior to and during the process. We know that we can use a cell box to transport you from A to B, et cetera, et cetera, et cetera. More things than I know. And so that is where I think we as participants in this test bed really want to see it go, right? The test bed for us was an investment. Let's be very honest. If you did not put equipment in there expecting a return, then I don't know what the hell you're doing, right? The test bed was an investment to showcase our products, not only independently, but collaboratively, right? We could have shown it in our place by ourselves. We don't need to truck all the way down to North Carolina to do that. The objective was to have equipment that would work within our system that researchers would have access to use to say, this is the process and protocol I want to use with little risk, almost no investment, and then decide to go buy our stuff, right? And I think that is what we've got to do a little bit better job of. So I presume later in the day we'll have more time, but from Biospherix point of view, we are thrilled to be here. We are getting ready to invest further in our presence down here in the not too distant future. We had an incredible discussion yesterday that some of you will hear more about later for workforce development. So we are invested, we are participating, but we need to see a return out of our investment down here, as I presume most of us do. So we'll talk more about that later, but that's the Biospherix concept for the day.
We can bring up Ronak now, if you're
yeah, absolutely. And Ahmed, thanks for the lead in because you show up on the first slide. So appreciate the shout out. Ronak has actually been developed out of the need for the ability to produce better, more, and higher quality cells and biologics. So in that first slide, we've had two great experiences so far on the test bed. One, as Ahmed mentioned, working with Biospherix in the in vivo, and you can see it in this two of the three pictures here, but also, as mentioned earlier, working with the externs. It really helped us hone some of our protocols. So how do customers read them? So what do we do? What you see on the right is a 1 centimeter cube hydrogel. This is the most advanced mass-produced hydrogel for cell culture on the market today. Every one of those little dots, there are thousands of channels that run throughout that 1 centimeter cube. What that allows us to do is take anything, whether it's an immortalized cell line or a PDX human biopsy sample, place it into the block, whether by pipetting or punch out, and now we can replicate the physiological model or microenvironment. We think we're somewhere between in vitro and in vivo, and we are calling that in similari. So it gives you a cost-effective way to scale. To give you a real-life example of that, we grew some PANC1 cells in 2D on the left, and we got a pancreatic biopsy on the right. And in the middle is what we grew. We have now done this across multiple cancer panels, showing that what is replicated inside the block looks an awful lot like in vivo. And we're able to expand that. Where you take a biopsy, it's very difficult to expand that. A couple months ago, we took a 15-day-old mouse and we took one of the whole kidneys from the mouse, put it in the block, and it expanded. We took the other kidney, we made a slurry, and that expanded inside the block too. So we can replicate that environment. What's this mean for the industry? Well, on the bottom, this is how drugs are, in this case, would be a government deployment of materials, but drugs are made virtually the same way. We start in 2D, we scale up, we have to produce a lot of cells, we see lack of physiological relevance, we have limited scalability. If you're making viral vectors, you know there's a huge limitation working with CHO cells to do that, inconsistency and expansion as we move from vessel to vessel. On the top, we have the ability with the BioBlock to do all this within that 1 centimeter cube. We also know that we can scale from a single experiment up through full production. With that, because we produce everything in-house and almost everything is made in the United States, we changed the form factor from that puzzle shape to a chip. We have an agreement with TPP now that we can put it in their tube, and all of a sudden we can create a small-scale bioreactor, create large-scale large-scale production in small-scale roller bottles, or we can create a perfusion method. Now we can essentially do whatever production model you want and however you want it in a much smaller rate. To give you an idea, when we're looking at this, that little tube, we estimate that we could get 1 billion cells in a 50 ml tube, which means your media production is going to go down significantly. Your space production is going to go down, reducing your materials by about up to 90%. And so the future of lab, thanks to an AI-generated model here, is what could we do? Ahmed, what if that was all a Biospherix environment inside there? You know, what could we do? It's. Are you in? Did I hear you say you're in?
Yes.
All right. Well, the future is unlimited. Right now, we are in the process of looking at some scalable deals. And we believe the first 2 are coming through the door right now. We just are looking to finalize paperwork and move that forward. So we are interested to talk to anybody if you're looking for a better way and higher quality way to produce your cells and biologics. Thanks for the time.
Well, hello, everyone. I'm Casey Clark. I'm with Phase Holographic Imaging, and I'm going to talk to you about our imaging mode for quantitative phase imaging with the slant and angle of cell quality control. Our technology works a little bit differently than most microscopes. Instead of looking at the cells directly, we actually use 2 fairly homogeneous beams of light to collect an interference pattern. You can see, 1 second here, let me get this going. You can see the beam comes in, it gets split, it'll pass through the sample, and then this undisturbed beam recombines, and we get an interference pattern like this. That interference pattern is unique to the cells that it passes through, so that when we reconstruct the image, we get a 3D model of that image. And it's a little bit different than doing like a Z-stack. You get sort of different information out of it. And a great example is right here, where these are some cells that we image, probably HeLa cells, and that's what your cells look like when they are in your culture plate. And with our technology, you can measure the height. We have a little bar right here, so you can measure the peak height of those 2 cells. And beyond measuring their volume and height, you can track them. So you start out with just a few cells, and you can color code them to the families and watch how they divide. You can watch this in real time. You can watch this in time lapse over multiple days or even weeks. And you can see how your cells divide and move in response to different stimuli or growth conditions. And that kind of leads into the smart manufacturing project that I'll touch on a little bit later. We can see applications for this technology when it comes to large-scale manufacturing, where you would have some sort of bioreactor with your cells in suspension, and you can feed that through with a peristaltic pump onto a microfluidic slide, which is then installed or worked into the Hall monitor. And so as that pump moves your cells through, you can get volume readings, cell density readings, and other viability metrics to see just how well your cells are growing. And if they're growing poorly, maybe you could have some informed decision from our quantitative information on how to adjust your manufacturing settings or the environment with Biospherix. Change the temperature, change the oxygen concentration, whatever you need to get your production back on track. Another method that you can employ this technology is what we're using right now with some regulatory science projects with the Navigator and Stephen Sawyer's team, where if you have a bunch of different types of cells or batches of cells, let's say, you could have 4 batches of cells going, and they're growing on your traditional culture vessels or maybe those like multi-stack plates, cell factories. And if the conditions are same between the cell factories and the plate, this is what would be called a sentinel plate. So you can watch those cells grow, and you can make a pretty strong assumption that what's happening on the plate is happening in your large-scale culture. And this would be for adherent cells. And again, you would have that informed decision on how to change the conditions of the production. And this is some work that we're building upon with, and I reproduced this image here with permission from Dr. Bauer. This is some of his work that he did with mesenchymal stem cells. And that leads into our smart manufacturing project, where we're trying to do something similar to assess the quality of mesenchymal stem cells and sort of break them up into different families, just like Dr. Bauer did, to show this is what a good quality stem cell looks like. And you can do that for whatever other type of cell that you want. If you want to look at pluripotent stem cells, if you want to look at muscle cells, whatever it is, we can use this technology based on their morphology, non-invasive. You don't have to take out samples and break sterility. You just do it in line, and you can get quality metrics out of your production facility. Kind of our vision, where we would look at the cells at extraction to make sure that we got a good batch. We can look at them during transport to make sure and ensure that your shipping conditions with cell box are optimal, to make sure nothing is interfering. And then again, right before you administer it to the patient, to make sure that your cryopreserved cell product is what you say it is, and the patient gets the best treatment that they need. Practical implementation of this technology is pretty straightforward. Our unit can fit into any sort of standard incubator or ex vivo unit. And that brings us to the smart manufacturing project that we have going on with Biospherix, QIAGEN, and SAS, where we're tackling that MSC quality control. And that's kind of what it would look like up in the test bed, which some of you will see later. So I thank you for your time. I thank you for putting this on, and I hope to see more of y'all soon.
Well, thank you, Casey. And let me see. I think that next we have Brinter. You do stop share. Brinter, do you want to?
Hey guys, can you hear me?
Yep.
This is Matthew Brovold. I'm at a really busy conference right now, but I'm trying to find some time to find a quiet place to chat with you, everyone in the incubator space. So I'm Matthew, CSO and co-founder of Brinter. We are currently working on a 3D printed orthobiologic for rotator cuff repair. So we've been in the test bed space now for about 2 years, and part of our plan, other than making this medical device, is to also manufacture medical devices or manufacture biologics. My printer is the only modular printer on the market. I can print anything from liquids to viscous liquids to plastics. So we can do the whole gamut and array of bioprinting and 3D printing in a small form factor. So we've done we're chatting with Biospherix now, getting our unit into the Biospherix device so we can have an entirely enclosed ISO 5 system. So if anyone is looking to print anything, prototype anything in that enclosed ISO 5 clean room, please feel free to reach out to me. So we're, kind of have a dual business model of making this medical device ourselves, and then also looking for future projects to work on to also manufacture in the space. So that's my 30-second pitch. Let me know if you guys are interested. Love to chat with you.
Thank you, Matthew. And then I think after Brinter, we can have QIAGEN, if you want to.
Well, yeah, Stephen, everyone, thank you so much for having us. My name's John Grebbins. I'm with QIAGEN. I believe we are new to kind of the REMDO organization, and really looking forward to kind of meeting all of you, working with all of you a lot closer. But just as far as QIAGEN goes, so our goal at QIAGEN is generally just making improvements in life possible. So again, I wanted to thank kind of the REMDO group, as well as those startup companies that we hopefully can be working with within the test bed. So in regards to QIAGEN, our whole idea here is to have a really focused portfolio targeting many different segments in pursuit of helping to provide answers to biological questions which organizations across the globe are trying to solve. So we're doing this through discovery and translational research, really providing tools for research from the bench to the bedside. We're also working with pharma and biopharma companies to develop companion diagnostics, essentially when they have these novel therapies or medicines, to create assays and such, so they can use these in order to diagnose those folks that should be treated with these particular therapeutics. We're also working in the clinical diagnostic space, enabling diagnostic tests so patients can get the treatment that they need. And lastly, we're supporting researchers who are bridging the gap between health and safety using molecular biology methods. So what does that look like? So essentially, QIAGEN provides a sample to insight workflow. And the whole idea here is to help researchers extract and analyze any sort of nucleic acid. So that's DNA and RNA. So any biological sample that's pretty much out there, we can extract DNA and RNA from it using our kits as well as instrumentation to automate and scale this. And all of you kind of talked about scaling, and that's what we're built for. We have a number of instruments so we can kind of scale research on the bench. So we've got a number of different trusted detective methods as well. So things like polymerase chain reaction or PCR. Everyone heard about this during COVID. And then we've also got qPCR, quantitative PCR, digital PCR, and then next generation sequencing or NGS to look at the sequences of those nucleic acids. And in doing so, we get a lot of different bioinformatic data, and using our QIAGEN DNA Insights group, Cutie, we can kind of analyze all of those large data sets and get valuable molecular insights. So we've got a team of field application scientists that can come on site, do training for any of these instruments, as well as kits that we have, and really just ongoing support for those founders and those scientists to continue developing experiments. So really, I mean, overall, at QIAGEN, we are addressing applications that closely align with REMDO and kind of those folks that'll be working the test bed. So one thing I do want to kind of drill into is our digital PCR system. So QIAGEN developed a digital PCR system that's perfect for companies working in the regenerative medicine space. So far we've placed around 2,700 of these globally. And what this does is it provides, as opposed to qPCR, which is kind of relative quantitation, we provide absolute target quantification of these nucleic acids. So we have a high tolerance of inhibitors when we're doing this digital PCR method, superior precision looking at small fold changes, increased sensitivity, reproducibility, which is huge in the scientific space as far as when you're doing bench research. And then lastly, we can do higher order multiplexing, looking at in a single sample, looking at up to 12 different targets, genomic or transcriptomic targets within the well. And we built this to scale. So a lot of you talked about scaling, and it seems like we have really complementary technologies. So we have 3 different versions of this digital PCR system: a 1-plate, a 4-plate, and an 8-plate system. So essentially, as different groups want to utilize this system, they can go in and continuously load into this 4-plate or 8-plate system, even if they're using multiple different types of targets, different types of plates. We can support that. And lastly, because we did build this to scale up to an 8-plate system, loading 8 96-well plates at a time, doing digital PCR, as the test bed scales up and you get more folks interested, we can scale with you. Another division within QIAGEN is our QIAGEN DN Digital Insights Solution Division. And this is essentially our bioinformatics group. So in our bioinformatics group, we develop tools that are driven by over 20 million scientific findings. And when I say scientific findings, I mean publications, within the scientific literature. And within this, we've done an analysis and aggregated these data from over 600,000 samples. The whole idea here is when people are doing biological research, we want to, in looking at large sets of biological data, we want to save those companies time as well as resources. And lastly, the whole idea here is QDI enables biomarker and target discovery to drive new innovations within the biotech and pharma space. So here's an example of essentially some approaches that these QDI tools or bioinformatics tools use for biological discoveries. So first, we can identify and prioritize molecular candidates. So we can explore thousands of curated studies that are already in the publications and hone in on the candidates of genes to reveal patterns of expression across different tissues and disease types. We can also look at RNA-seq data. So compare the RNA-seq data being developed generated in the test beds to thousands of curated projects that are already in the literature, and compare that RNA-seq data across tissues and disease types to give you those insights that these labs are looking for. Also looking at differential gene expression. So we can identify dysregulated pathways within these different in vivo and in vitro studies, looking at gene networks from your own data sets as well as published data sets that are out there. Oops. And we're also looking at biological, we can allow biological network analysis. So QDI will allow you to kind of see how disease progresses through a network cascade, as well as you can explore data against high quality, manually curated molecular interaction networks. And using these particular platforms like Ingenuity Pathway, we can kind of uncover potential mechanisms of action, identifying new targets, and even repurposing drugs based on the literature using analytics and artificial intelligence. And last but not least, we can explore molecules and pathways associated with diseases. So we can discover relationships between molecules, drugs, targets, diseases, functions, as well as toxicological processes within cells and animal models. So this is kind of the QIAGEN team that would be supporting Remdo. We really look forward to working with you. Again, my name is John Grebens. I'm the specialty sales manager for digital PCR, but we have a whole team to kind of back the Remdo and testbed. So thank you so much for your time, and please feel free to reach out if there's any collaborations that you'd like to explore, or and just in general. Really looking forward to connect with you all.
Yeah. Good afternoon, everyone. Thanks, Stephen, for having me. My name's Joe Rector. I'm the field applications specialist at Sartorius. Like Stephen mentioned, I'm going to be talking about the IncuSite, which is our live cell imaging and analysis system. So really, it's a powerful tool that's helped change how researchers study cells in real time. So we're probably one of the first to go out and do real-time imaging of cell cultures. And so unlike traditional endpoint assays that might only provide a single snapshot, the IncuSite will do continuous monitoring or capturing those images over time, collecting that data and allowing you to monitor that cell behavior dynamically. And that's without ever disturbing your cell cultures. So we believe that this approach is going to be able to provide deeper biological insights, improve reproducibility, and accelerate the discovery pathline. So let's begin with some of the hardware components. The IncuSite is designed to sit inside of a standard tissue culture incubator. That's going to help ensure that your cells remain in an optimal environmental environment to maintain those optimal conditions throughout the entire course of your experiment. In terms of imaging, we have bright field capabilities, phase contrast, and we can image up to 3 fluorescent channels at a time. So that's going to give you the flexibility to track multiple parameters in a single well or assay. Now, the system supports a wide range of plate formats. That's going to go from 6 well up to 384. We also support slides, flask, dishes, and that's going to help you increase your scalability as well as the flexibility. With the automated imaging aspect, that's going to help eliminate any variability that you might encounter through manual microscopy. And we also have built-in software that helps streamline that data analysis. That's going to reduce hands-on time and increase efficiency. In terms of the key applications, the IncuSite really shines, I would say, with our spheroid and organoid applications. Those can be found on the systems within the test bed. So as 3D models become a little bit more widely used for drug discovery and disease modeling, the IncuSite keeps up and provides real-time tracking of growth, viability, and the response of those 3D cultures to treatment. We can do that as a single spheroid scanning method. So looking at a single individual 3D culture there, that's going to be really great for tracking morphology and viability over time. We can also do multi-spheroid models. So looking at smaller spheroids per well, looking at population level changes. Of course, that's going to be great for cold culture models as well. So with our 3D capabilities, we can track changes in size, density, or fluorescent intensity. And that's going to give you a different ability to capture those quantitative assessments of maybe drug responses that you wouldn't normally get with the traditional method. We can also do migration assays. So whether those are scratch wound or chemotaxis. With scratch wound, we have a system that creates reproducible wounds in there. We also have a special metric called relative wound density. That's going to be different than typical simple gap closure measurements. That relative wound density helps normalize your data based on the initial wound size and density, allowing you to determine if your cells are proliferating within or migrating down from a non-wounded space. One of the last applications that I'll discuss is our AI cell health module. So traditional cell health assays are going to rely on endpoint measurements that might miss those changes throughout the course of your experiment. The IncuSite obviously overcomes this by doing continuous monitoring, but also by applying deep learning algorithms to continuously assess morphology, proliferation, and death in real time. So for example, that AI-powered segmentation, it's going to be able to give you label-free counts, as well as distinguishing healthy from dead or dying cells. It's going to do that through subtle morphological changes, and that capability can be really useful for toxicity screenings, immunotherapy studies, or time course drug response experiments. So why choose to use the IncuSite? How can it help simplify progress in the lab for you? Like I mentioned, we have unintended long-term monitoring, so hands-off, and it's going to increase reproducibility by having that automation. You'll get real-time kinetic data, so you don't have to worry about missing any endpoints as you would with a more traditional method. We can multiplex, so we can do multiple channels at a time to get more readouts in a single experiment. We employ AI, so again, for automation, to remove biases, to simplify that data analysis. And then lastly, we have optimized workflows. So some of the applications I mentioned, those will all have product guides, protocols, really making it streamlined and easier for you to take advantage of those tools that are going to be really beneficial for oncologists, immunologists, cell drug discovery labs. All of those will be able to benefit from the IncuSite. So if you have any questions, maybe at the end or next week when I'm back on site, I'd be happy to take those, but I'll pass it back to Stephen, and thank you again for your time.
Excellent. So I'm Vicki Martin. I'm our VP of Research and Clinical Science. And let's see if it'll advance my slides. There we go. Okay. So our technology is called the Multi-Tissue Platform, or MTP. And we're in a little different space than I think a lot of the folks in the test bed. Our material is a decellularized extracellular matrix-based material, and it's made from pig spleen and lung. So ECMs are not new to the wound healing and regenerative space. Where our material is different is that it's sourced from multiple tissue sources and specifically designed to contain less collagen and more of the other biomolecules that are important for wound healing and a shift to a positive regenerative response. So, the material contains collagen, elastin, fibronectin, laminin, glycosaminoglycans, all those structural components of the extracellular matrix, as well as hyaluronic acid, a wide variety of growth factors, and exosomes. And we've been on the market since 2018. So our initial wound powder has been, we have tens of thousands of clinical use cases with the material with an excellent safety profile, no adverse events reported. And then just last year, we received FDA clearance for the first ECM-based gel or liquid material, which is what we have in stock at the test bed. And so that's MTP gel or RegeNeraTOR. So at a high level, how the material works is it promotes regeneration in kind of two key ways. First, it's providing those physical structural components that the body needs to regenerate tissue. So the collagen, elastin, like the physical building blocks that the body needs. But then those exosomes and growth factors are recruiting stem cells, immune cells, and the other cell types that are important for an appropriate remodeling response. And so what we see in the clinic is superior performance in terms of getting wounds that are stalled in that kind of chronic inflammatory state, move them forward into a positive healing response with less inflammation and a decreased scar or fibrosis formation. Another side kind of project that we have going on is during the routine testing that's required for FDA and our viral inactivation testing, we found that our material has innate broad spectrum antiviral activity against envelope viruses. So this is a whole new aspect of our material that we've been looking into. I've got listed here on the screen the variety of in vitro tests that we've done, and there's slight variations from one virus family to another, but we show a consistent, generally log 3 to log 4 reduction in viral load with a 1-minute direct contact exposure. So this is kind of exciting and a new avenue of capability for our material that we've been looking into. So at a high level, in terms of how the material can, some of the advantages over other ECMs is, like I mentioned, tens of thousands of clinical uses to date with no adverse events. So the safety profile is really well established, and it has been used alongside a variety of other therapeutics. So we envision that this could be an add-on to a lot of other treatments. It's very versatile. The stability, it's super simple. It's both the powder and gel forms are provided sterile and they're shelf stable. There's no specific temperature requirements. They have a 2 to 5 year shelf life so far, and we have continued testing to extend that shelf life. And it's super easy to use. It's literally like open the package, apply it to the wound. So currently they've been used in topical wound applications, but especially with this new discovery that we have both tissue regeneration and antiviral properties in a single product, we think there's a lot of untapped potential here, and we're excited to move things forward. So where that leads us in terms of collaborations with Wake Forest and the test bed is we have a current FDA cleared powder and gel product. The gel is in stock at the test bed right now. We can get powder, but I think the gel is probably a little bit more versatile for most people to use. And we're working on new formulations because we really want to take this from a topical wound product to other areas of the body and other disease states where stimulating regeneration and modulating inflammation would be important. So I've got a list here of a few of the future formulations that we're currently thinking about or working on. And what we're really looking for is research partnerships. So there on the screen here, I have a whole bunch of ideas that we have for future applications of the material with some of those new formulations. But knowing some of the work going on at Wake Forest, I am sure that there are other places where this type of material would be valuable that we haven't even thought of yet. I do also think it's important to note that in addition to being a standalone therapy, we see a lot of value in combining this with some of the cell therapies that are under development, because it would really put those cells in an ideal environment to enhance their performance upon delivery. So imagine taking your cell therapy and delivering it in a solution of all the other components and growth factors that are needed for the ideal regenerative response. I went ahead and left this slide in here at the end because I wanted to mention that between the regenerative capabilities and the antiviral capabilities, we see a lot of potential for DoD type applications, which I know is a big thing at Wake Forest and in the engine. And I also think I just threw in one clinical case, I think, as an example of what we've been seeing in the clinic. So this was a patient with severe spreading necrosis in the shoulder, and it got down to tendon and bone. And using our powder material, we were able to not only heal the wound, but also you can see there was significant regeneration of that volumetric tissue loss. And this is pretty representative of what we're seeing in serious trauma cases. So it's pretty exciting, and we are eager for collaborations. So this last page is just to it's a little bit of alphabet soup, I think sometimes. FetTech is our parent company, but we've established subsidiary companies for each of our use applications. So these each have their own website with additional information. So StemSys is our original wound powder Rheamid is our MTP gel, and then Viramed is our development of the antiviral side of things. And I would be happy to talk to anybody that's interested in potential for using our product. But like I said, we have MTP gel. Stephen has a stock of it at the test bed right now. So we'd love to collaborate.
So while I'm pulling up my slides, my name is Bobby Atkinson, and I am part of the Apridia team. And I'm actually located in Greensboro, North Carolina, and I'm joining you today from our Kalamazoo, Michigan facility. And we actually have a pretty good local team. Not only am I located in North Carolina, but Bradford, who's in the room, is local, and Fukumoto Takanori is also in North Carolina. So a little bit about us. We're an end-to-end pathology lab provider. So this is where our core business is today. So if you're processing tissue using these consumables or using this equipment, it's likely in your lifetime that you've heard of brands like Thermo Fisher, Richard Allen, etc. Those are all companies that have the Apridia legacy. So what has happened, even though we have origins of all these companies, Apridia formed as a result of PHC Corporation buying this division from Thermo Fisher Scientific in 2019. So you may see Thermofrost Plus slides or something like that in the lab, and you're like, oh, who makes that? And it's like, hey, that's Apridia. So the name Apridia hasn't been out there in the market for that long, but our legacy brands definitely have. So we have some of this equipment in the test bed. If you're interested in learning more about what we can do for you from a core pathology perspective, happy to get you in touch with the right people. But I also want to touch upon is we're part of a larger corporation, which is the PHC Corporation. So we have freezers, we have incubators, we have refrigerators. There's other things within the lab where you may need help. We can provide that assistance as well. So if you walk around the different parts of this campus, you'll see these brand names as well in different labs, and that's part of the PHC family that we're part of. I think this is also a win that I would like to mention is we have a tissue processor called the Revos, and WFirm has recently helped us on some studies and helping us publish some white paper documentation regarding the Revos tissue processor and how it can be more efficient and improve performance versus other tissue processors, including our own. So this is definitely a win, and how we're partnering with the Wake Forest community to figure out how do we accelerate innovation for our own company and also make it more easily accessible for our customers. This is also something that's very interesting. It's a little outside the core pathology market, but we recently announced that we would partner with a company called Novascan in April of 2024, where a dermatologist could excise tissue in a Mohs procedure and put that tissue in this machine and put a electrical assessment through that and confirm that the tissue is cancer-free. So I think this is just an example of where we're branching out outside the core pathology lab to see what other ways can we help our customers and our patients make a accurate and quality diagnosis. So this is a pretty exciting product that we're proud of, and it should be subject to launch later this year in 2025. But now let's get into the exciting part, which is digital pathology. So as we started off with the big win for us with our clinical clearance, but not only do we have that clinical clearance, but we also have all these instruments, including the P1000, which is in the test bed today. So if you're a lab, you're scanning slides, we have a variety of different products that we can offer you, we can demo. I failed to mention in the intro here is we actually have an office on the first floor of WFirm. And so we have the ability to talk to you and also help you if you're interested in any type of digital pathology scanning. So bright field fluorescence, different objectives, water immersion. We can do all kinds of different things, both in the research and clinical space. And so, yeah, we're really excited and we're really thankful that Wake Forest partnered with us on our clinical study. It was a lot of work by the team and also with Brad and Fukumoto-san to make it happen. And every day for like the last few months, we've been asking, oh, when's it coming? So on March 4th, when we were able to announce this, is a big win for not only for Apredia, but also for WFirm and that collaboration. So if anyone has any questions about the E1000 DX scanner, we're happy to talk to you or your partners about how you could use this equipment in your clinical workflows, wherever that may be. So yeah, it's pretty exciting. So a large flurry of activity going on here at Apredia in regards to this FDA cleared device. So again, just thank you to the Wake Forest community for helping us make this realization for us.
I think this panel is going to be talking about a kind of larger ecosystem opportunities that the testbed intersects with.
This next section, we wanted to talk about additional ROIs and expanding some of these opportunities. So first on deck is Nancy Johnson. She is Executive Director of the Piedmont Office of NC Biotech. And Nancy, you can take it away. Well,
fantastic. Thank you all very much. Thank you to the organizers. Thank you to the presenters. What an informative and important discussion that's underway. Yeah, I don't know if I can see everyone, but just in the spirit, yeah, I see. In the spirit of transition, if you are near a window and can look outside, and if you're in North Carolina, taking the sunshine and the blue sky, stand up if you're able to and stretch, take a drink, a snack, whatever helps you transition. Just let's pause for a minute so we can refocus. But I just wanted to tell you that I'm always blown away when I see innovators with this intense interest in advancing new technology and ideas and solutions. And I get really excited when this is happening right here in what is known as the Piedmont Triad in the center of North Carolina in the life sciences ecosystem. So I was introduced, Nancy Johnston, Executive Director for North Carolina Biotechnology Center here in the Piedmont Triad region. My organization is based in Research Triangle Park. I think we have some partners on the line that have a global footprint and a U.S. footprint. So what I'd like to do today is just create some context on my organization and just life sciences in North Carolina around that topic of ROI. Talk briefly about some resources that you may or may not be aware of, and this will be pretty streamlined, top line, just to empower you, and then some quick save the dates to make sure that you're aware of some things that could be relevant. So let's get started. I know I just have a few minutes here, and I will go ahead and share my screen. Okay, what I'd like to do is talk a little bit about the why, and I think this is incredibly important based on what I've heard from everyone, evidence and opportunity of North Carolina. So for those who may not be aware, we just came out with a Techonomy report, and so North Carolina's life sciences employment now exceeds 100,000. So that's a new milestone. You can see the concentration of areas across the U.S. And this is a report that is available on our website. For those who want to understand where we fit into the scheme of outpacing national industry growth, we're second only to Massachusetts. And I also want to encourage everyone who is engaged with the engine and the test bed to know that you are in the right place at the right time, and you helped contribute in part to some of this percentage growth of establishments within the state. Our life sciences employment continues to outpace national industry growth. I think I mentioned that to you, and I'm thinking that these are not moving forward. Here we go. So it was so important of a point, I thought I'd bring it up again. Wages are always important. Just want to create some context of the importance of the industry. I know we're going to hear from some partners around workforce shortly. Economic impact in North Carolina is significant, $82 billion. And I want to pause here for a moment. This is the state of North Carolina. We are in the center of North Carolina. You can see where our ecosystem and subsectors are represented, where you can see the pharmaceuticals to the research testing, med devices, ag, and even just bioscience-related distribution. And what I wanted to note to you is the strength is not only here at the core with the engine, with the test bed, but it also reaches out across the state, which is a really viable ROI for our great partners. Some areas of strength for North Carolina that I think play well into what was just heard about these companies and their advancement. It's around biopharma manufacturing, contract research and testing, research and development, and of course, ag tech. And this is where I'm going to spend just a moment of time because this is partly what I want to deliver to you on the ROI with my comments. Our programming does lend itself to supporting innovation and job growth in the life sciences. Our vision is North Carolina as a global life sciences leader. So specifically, resources that could be made available to you to raise your visibility. There's some basic attributes that can be found and resources on our website, which is ncbiotech.org. But I'd like to extract that just for a second. We have a company directory. And if you are not listed on that company directory, that would be a first step in evaluating whether you would be eligible and whether we can get you listed. Secondly, we do have the opportunity for job listings where you may have some vacancies. It's going to raise your visibility as a company operating in North Carolina and seeking talent. It ranges anywhere from an associate's degree to PhD scientist. We have information on our website on incubators and research parks. You're about to hear from the Innovation Quarter. We also have networking and event opportunities. We actually have event grants. So I would say pay attention to that calendar and opportunities to network there. We have something that might be of interest to some of our what we'll call service providers that presented or are participating on this call. It's a baton referral network where we'd want to talk with you and see if you are wanting to support early stage startups, what that could look like, and maybe that could be increased visibility for you. We do have a life science intelligence group, which is expertise and resources for life sciences market research, competitive analysis, business development, and that is something that we might want to consider plugging you into. So I know that's a lot, but I did want to just raise awareness for you about those potential resources. And then lastly, I did want to save the date for you. We, NC Biotech, will be participating in Bio 2025 in Boston. We will have a North Carolina pavilion. There is some opportunity and availability to come and join us. I know Brinter, I believe, will be there. I think the Innovation Quarter will be there, and that is June 16th through the 19th. We will also be working in North Carolina to celebrate Life Sciences Week, which should be mid-September, so there'll be opportunities there. And lastly, my office works with our Regional Advisory Committee. I think Russ Reed is in the room. He's a member. And also Ginevra Kelly with W Firm to organize what's called Triad Bio Night. That will occur in June of 2026. So I'm very happy to talk to you about these iconic milestone opportunities to increase your visibility. So thank you very much.
So next up, we have JT Warren. He is the Economic Development Analyst at Greater Winston-Salem.
I don't stand behind them.
Yeah, I'm gonna turn it around. Awesome. There you go.
Thank you. It's a super unflattering angle, but we're gonna make it work. There are two conditions to. Are we good? Thank you. I appreciate it. Two conditions to this assumption. I think I might be the only one in here with just a bachelor's and no patent to my name, but that's okay. Okay. Thanks, Karen. Thank you. But my name is JT Warren. I'm with Greater Winston-Salem, Inc. It's the organization that combines our Chamber of Commerce and Economic Development. So I work alongside Isaac Perry at Innovation Quarter and help find landing spots for companies who are looking to relocate or expand in Forsyth County. By no extent of the imagination or your imagination, whoever's looking at me right now, am I a scientist? But I can definitely speak to you about the importance of the test bed in Innovation Quarter's growth strategy. So the test bed is a microcosm of Innovation Quarter. And it started decades ago and it continues into the future. It's kind of like a spider web where you have lots of different things culminating to one point, and then a lot of different connections to those different strands. That's helping support lots of different types of companies. The testbed offers product and process collaboration for companies, researchers, and developers alike, as well as approximately $1 billion worth of benefits to those members that are part of the testbed. So you heard from a lot of the members today. Many more are around and are upcoming, I'm sure. So thank you guys so much for your time, for your energy. And if you guys have any questions for myself or for Isaac Perry, feel free to reach out to Stephen and he can happily connect you guys to us. So thank you guys so much for your time, and we look forward to seeing you guys around more often. Thanks.
So next up, we have Russ Reed. He is the Executive Director of the National Center for Biotech Workforce at Foresight. So welcome.
Thanks, John.
We're going to hear about workforce development and training.
Yeah. Well, good afternoon, everybody. It's a pleasure to be here and to see you all and to meet this group. Actually, it's really interesting because we were in the test bed last night, and we were with Ahmed, and he has a vision. But I want to tell you a little bit about Foresight Tech and how it's connected to probably your city. So if you're online, we're probably connected to you. Let me just tell you about that. So we are a community college. We're approximately 2.5 miles away from here. We do have a downtown campus, so we are in the Innovation Quarter. We have a lab which perfectly duplicates what you have here at Institute for Regenerative Medicine. It's part of our national center. I think the rationale for wanting to deal with us is because we are a national center. And what that means is we're connected to 130 community colleges and universities across the country. In fact, we meet weekly. So we're able to discuss key issues that are going on with respect to knowledge, skills, and abilities for the technical workforce. Many of you have interacted with us. You know that we're very strong in our program in doing professional development. Where's Katie? Katie is our star graduate, so she can speak about the academic program. I'll talk about the professional development program. But if you've seen the kind of talent that has come to WFirm, there are many people. Whenever you take a tour within WFirm, you're bound to run into a graduate from Foresight Tech. And the reason, the simple secret for that is because we design our program with industry and for industry. So what we do is we go through a skills analysis with you to find out what are those emerging skills that you need to know, that you want to have in your workers. So we're as I said, we are the center. If you go to our website, which is biotechworkforce.org, we are the center who developed the first ever global, I'm told, by our subject matter experts, medical device skill standards. So we always go back to our medical device skill standards, and also we develop the core bioscience skill standards. So if you take laboratory, biomanufacturing, medical device, and you melt them down to the core, that's us. We did that. That's our work. So we're very proud of that work. We also most recently teamed up with Solano College and also Montgomery College out of New Jersey, or sorry, not New Jersey, Pennsylvania, and we develop cell and gene therapy skill standards. So if you go to this website, biotechworkforce.org, you're sure to find, if you scroll it down, you'll find all those skill standards that come out. They're nicely done. They're done professionally, and I think you'd really enjoy them. Let's just very quickly talk about some other programs we have going. So a lot of our financing comes right now. We're getting some financing from the engine. We're very appreciative of that. And let me tell you what we did with that financing right away. We knew that we did not have a bioprinting system at our college, so we have purchased one. We also knew we did not have a scanning electron microscope, so we purchased one. We have a scanning electron microscope which fits on a desk this big. We have a bioprinting system which fits on a desk this big. But the importance of that is now our students that go through Foresight Tech and take the biotech program are perfectly aligned to what happens within WFIRM. And I think that's great because that matches up to the test bed very nicely. The tour yesterday showed me that. So I was really kind of pleased about that. As you know, we are working with the engine. So my counterpart, Joan Shanks, online. She handles the education part. She's Chief Educational Officer for WFIRM. So we have worked together for 15 years. We know each other's work. And what we try to do is make sure we work in synergy. So that's very important. I do want to tell you very quickly about an NSF program I have. I know that Stephanie has advertised it within the engine and within the ecosystem. But we have a program where we take fellows, we call them fellows, they're community college instructors from across the country, and we place them into local companies for a period of 8 weeks. We pay the freight. Think about that. We pay their airfare, we pay their hotel, we pay them, and we pay their transportation locally. And we give them a stipend so they're able to provide for food and so forth. We do that. We've done it now for the past 3 years. This is our third year of doing this. This program has been extremely successful. We've had people in Pfizer, we've had people in WFIRM, and we've had people in Carinetics here. We've had people in Novex. And so I just want to let you know that I have one person who is not placed yet. And so if you want to take, if somebody has a summer project they want to do, 8 weeks, starts in June, flexibility your way, they're your employee. We make a contract with you, but the NSF, thank you, pays them. It's perfect. Great deal. I'm going to stop, and I'm just going to stop now because Ahmed has a program that he designed in his head. He was dreaming one night about the test bed. And so he came to us yesterday, and he had a few educators in the room, and Karen was online and everything. And I think it really is a great idea for the test bed, for the area, and I think it has a lot of potential. So, I'm not going to try to parrot what he wants to do. I'm going to have him come up here and tell it. Come on up there. Yeah, please. Yeah. The short story, right? Yeah. Thanks very much. Please partner with us. We're there, Foresight Tech, biotechworkforce.org.
Is this okay, or do you want me to do it afterwards offline so we get the last two done? Because I'm happy to pop it in later.
So, do you want
briefly
2 minutes?
Sure. So thanks, Russ, very much. I mean, we had an invigorating meeting yesterday with Carolina University. We didn't get Salem. We got Bryant and Stratton College out of New York. We had Foresight Tech there. And a vision that we're putting together is to develop a curriculum that Josh helped draft yesterday in a few minutes that will allow students to come into the test bed and get certified in a variety of our products. So again, as I've said multiple times, every time I come here, this is not about Biospherix, it's about the we. And so we developed a straw man yesterday that included a variety of companies that we were familiar with, so it was kind of easy. But over time, we expect this to grow and include many, many companies, if not all of the companies in the test bed. And the short vision that Russ alluded to is that you will basically take what went from a certificate program to now a 7-week curriculum where students in the local colleges, both community colleges and four-year colleges, can come in and learn how to work in an ex vivo system, how to work with media, how to grow cells, how to image cells, how to transport cells, and how to do a variety of other things over time. So I only have 2 minutes. We'll talk more about that. but I promise you by mid-April, you'll hear a lot more about it. And then we'll be looking for other participants to join in the second iteration of this. So thanks for us very much. Thanks, Doug.
Brilliant, brilliant. Oh, go for it, Joan. You're on mute, Joan.
Thank you, Ahmed, for that. That's really very exciting, and I look forward to seeing the straw man or the straw woman. But let's, even when we're building that out, let's not forget also our connections to the CTEs, which were really very much lacking in our ecosystem. And we're starting to partner more with the North Carolina Association for Biomedical Research through their curricula that they deliver to our middle school and high school students. The CTEs are connectors. So Ahmed, we can do an adventures and biotechnology program for high school students starting to get mini certifications or training on some of these technologies. I mean, this is exploratory here with all of us, and we have a lot of work to do ahead, but I couldn't be more excited about that. And boy, outside of that, also the quick presentations in the test bed. We all need you there for our public day. We're in one room because we're hoping to excite a lot of attendance, and we're getting folks registering from across our region to appreciate what we're doing, and moreover for the general public and students, for them to understand how they can sprout a career in this field and what these technologies look like, what the opportunities are. And also, you know, I saw a lot of young folks present from these companies. We have a lot of students that are interested in the entrepreneurial industry aspect. We need to get outside of just this room at times. So that's my plug. It's great to be here. Sorry, I'm actually not in that room. But anyway, that's it. That's all I got.